Welcome to the Pod of Inquiry, everyone. My guest today is Dr. Kevin Tracey. Dr. Tracey is a neurosurgeon by training, but he is now the CEO and president of the Feinstein Medical Institutes, which is a huge research consortium up in New York. The work that he has done has led to his distinction as being considered the father of bioelectronic medicine, which we'll talk about in this episode, and you'll get an idea of what that is and how that's going to play a huge role in the future of where we manage conditions no longer by pharmacological methods, but actually by vagal nerve stimulation methods. This is a fascinating conversation. This gentleman has more than 100 patents. He's considered one of the most sought after researchers in the world and has been cited so many times for all of the articles that he's published. So please enjoy this conversation that I have with Dr. Kevin Tracey.
Welcome to the Pod of Inquiry. Ready for some thought-provoking cerebral whipping and the destruction of ridiculous medical dogma? Want to shift those stale, moldy old paradigms? Well then, don that thinking cap of yours, get down in the cave, and join our host, Dr. Stephen Barrett, for this week's episode. Let's start spelunking.
Welcome to the Pod of Inquiry. I've got Dr. Kevin Tracey with me here today. I'm really excited to have this conversation with you, and thanks so much for taking the time to come on the show.
Thank you for having me on. It's great to be here. I'm looking forward to chatting.
Yeah, so you were kind enough to send your book down to me, and I started reading it, and I have to tell you, it was a wonderful book, and I would encourage about half of our audience is physicians, and about half are just people who are very interested in optimizing their health. And the way you wrote it is it just flows easy. It's understandable. You don't have to be a neurophysiologist to understand. I think you broke it down really well. So before we get started talking about the great nerve, which is the title of your book, and of course the vagus nerve, tell us a little bit about how you became the CEO and president of the Feinstein Institute, what y'all are doing there, and then we'll get into the more of the weeds, so to speak, about what your research has done and where it's going to go.
The Feinstein Institute is in New York. It is part of a health system called Northwell Health, which is the largest health system in New York. It sees about 2.5 million patients a year in nearly 30 hospitals that go from New York City all the way out to the Hamptons, all the way into New Jersey, Westchester, now in parts of Connecticut. So it's an enormous health system. We, some years ago, 20 now, took the research assets of Northwell and put them under the not-for-profit Feinstein Institutes for Medical Research. So now we have about 1,000 full-time employees doing research in about 100 laboratories. Most of the research in the laboratories is in a research building, but we also have 4,000 part-time researchers and clinical researchers that work all across the health system in a variety of specialties. Bioelectronic medicine, we'll talk about, molecular medicine, cancer research, psychiatry research, and we have expertise also in what we call health system science, which is data-driven outcomes research, and also in obviously clinical trials. So it's a big operation, and what holds it all together is a culture of collaboration focused on a mission of producing knowledge to cure disease. So it's a great group of people. I'm very privileged to work with them.
Well, it's really exciting. One of the things that I picked up in your book was where you talked about Sangeeta and what she said about your research. I'm just going to quote it here because I think this is so important, especially in all the research. And you said that she described your work, and she's the co-head of your laboratory. And she said, everything cannot depend on one protein and one cell type, right? It has to be the whole animal picture. So we don't study one cell type or only one disease because we are interested in what happens as a whole when different physiological systems are in play, and we want to find cures. And I think that actually resonated with me so much, Kevin, because so much of the research now is not translational, because it is only one protein. And then when they put these one proteins in or one enzyme of the pathway, it may work, it may not. It's not taking in the complex totality of human physiology. So I thought that was really to me, that was why your research resonated with me in such a powerful fashion.
Sangeeta is, as you said, the co-director of my lab, the co-head of my lab, and she's very, very bright, as you also just point out. And you're absolutely right. We've gone through an era in science, and it's a long story in the evolution of, or the history of science. But where we are today is, for the past 20 years or so, scientists have been rewarded for reducing complex problems down to minuscule single answers. And that works in terms of, if you want to solve the molecular structure of the folding of a protein, and you want to look at the individual sequence of nucleotides that make up the language of a gene, or the words, the encoding of a gene, that all works, and it's interesting and it's intellectually satisfying. But it doesn't mean you can go down from the granular level of highly reductionist science and make accurate predictions about life. Life is an emergent property. You can't. If I gave you the sequence of the human genome, you couldn't produce life. It requires other systems, and it's the interaction between systems that's always fascinated me and Sangeeta. And when you start at the level of an animal that has several organs that are all operating based on the dictates of specific cellular mechanisms within those individual organs, you can study an organ, you can study a cell, you can study the molecules in the cell. But at the end of the day, all the organs have to function in balance with each other.
Right.
And that's what we call homeostasis. And the nervous system, because of the way the mammalian life evolved with a brain and a nervous system connecting the organs, you can think now, and what we've been doing for the last 20 years or so is thinking about understanding physiology and understanding biology as a process of organs functioning in homeostasis, in large part because they're functioning minute to minute under the guidance of reflex circuits in the nervous system. So when the doctor taps your knee and your leg pops up and you said, "Who did that?" That's a reflex. That means that there's an input to the nervous system and the output causes your leg to go up. Well, that kind of mechanism is occurring in every organ in your body, every second of the day, all year long, so that your organs function in homeostasis, in balance, and you don't have to think about it. And that's what that model is what completely fascinates Sangeeta and I in the lab.
Yeah, it's really interesting. As I talked to you before we started recording, I've been focused on peripheral nerve for such a long period of time that you kind of get down in your own weeds a little bit. And we always would think about, well, can we reestablish motor function, and can we do something to improve sensory, whether they were sensory deprived or hypersensitive, but never really thought about nerve as a homeostasis mechanism for really what I would call, for lack of a better term, just overall physiological health, which I think you might have been the first person that kind of put this inflammatory reflex into conceptualization. And, you have to ask yourself, well, why don't we think of things in different parameters? But your work with rheumatoid arthritis and the cytokines is fascinating, because that's something that, coming from, traditional, more like, okay, sensation, motor strength, yada, yada, you don't really think of it in that parameter. And that, for me, was like, wow, I kind of had to like shake my head a little bit going, this is really very, very cool. So that's pretty astounding, So tell everybody how you kind of figured out this inflammatory reflex and what it is, and then we can go into the vagus nerve itself and get a little bit more, because I think you laid it out in your book extremely well about how it's very, I don't know if a nerve can be eloquent, but it's pretty eloquent.
Thank you. The inflammatory reflex is a reflex in the vagus nerve that connects the brain to your immune system and functions like the brakes on your car. It slows down inflammation, and that's the connection to rheumatoid arthritis. In rheumatoid arthritis, there's too much inflammation, And the drugs we have to treat rheumatoid arthritis are immunosuppressive. They have serious side effects, and the drugs are not effective in all patients. In fact, they're probably only effective in about half of patients. There's a recent survey with the significant dissatisfaction of many, many rheumatoid arthritis patients with their current drugs. So that's the link between inflammation and rheumatoid arthritis. And we'll come back to where we are now with the recent approval by the FDA to target this story using a computer chip to treat rheumatoid arthritis patients by targeting the inflammatory reflex. And you asked where it came from. Well, like many good stories in science, it came to us by surprise. We were working in the laboratory on developing a drug to stop inflammation in the brains of animals with a stroke. And the idea was if we stop the inflammation that accompanies a stroke, which is, of course, brain ischemia, then perhaps by reducing the inflammation, we would reduce the size of the stroke in these mice and rats. And in fact, we did that. And that's what we expected, less inflammation because we injected the drug directly into the brains of these animals, less stroke damage. What we didn't expect, and came as a complete surprise, was that when we injected the drug into the brains of the animals, it also blocked the occurrence of inflammation in the organs of the body of the animal. Now, there wasn't enough drug in the brain to leak out and saturate the organs, so something else was going on. And it really took many months to figure out that the signals were traveling from the brain to the body, the immune system in the body, in the spleen, for instance. And these signals were traveling in the nerve called the vagus nerve, and they were turning off inflammation. It took years later that we understood the molecular mechanism. The molecule we had made to stop inflammation was actually activating neurons in the brain, which are the origin of the vagus nerve. So perhaps I should explain what is the vagus nerve.
Right, please do.
The vagus nerve, you have two of them. We say the vagus nerve, but you actually have two, one on each side. Arises at about the level of your ear and the base of your brain, and comes out of your skull and travels down the neck on both sides, across the chest on both sides, and down into the abdomen. And along the way, it sends off branches that touch all of the organs that you never think about all day long: your heart, your lungs, your liver, your GI tract, etc.
Right.
So I said, you have two vagus nerves, one on each side. Within each of these vagus nerves, you actually have 100,000 fibers. Now, each fiber has a specific path, a specific origin and insertion, and a specific function. So you really have 200,000 vagus nerves.
Mm-hmm.
And that's a remarkably seldom appreciated fact, because, as I said, I'll repeat, each and individual fiber has a specific job, a specific function in reflex circuits that maintain the homeostatic function, the health of all your organs. So the discovery that activity in certain vagus nerve fibers could stop inflammation was quite surprising and quite revolutionary, because if you know that the signals in the fibers are like the brakes on your car stopping inflammation, then I realized, and I wrote this on a napkin in 1998 before I filed patents, we realized that you should be able to build a device to electrically stimulate those vagus nerve fibers and suppress inflammation, and do that in people with rheumatoid arthritis. And it turns out that we did, and you can, and it works.
Right, right. So let me ask you a question, because you're talking a little bit about the anatomy of the vagus nerves. Why is it that everything is approached on the left side, the left vagus nerve? Is that some for access, or is there a difference in its innervation patterns?
So yes to all the above. The historic answer is rooted in the regulatory development of the first vagus nerve stimulators in the clinic, which were FDA approved back in the early 1990s, in the late 1990s. These vagus nerve stimulators were implanted to treat patients with epilepsy, and then subsequently also got FDA approval for use in treating depression. Both cases, epilepsy and depression, in patients who had persistent conditions despite maximal drug therapy. And so in the early clinical and surgical experience of implanting those devices, there's two reasons that the left side is preferred. One is to minimize the risk of complications to the recurrent laryngeal nerve, which goes into the larynx.
Mm-hmm.
And another is to reduce the complications of arrhythmias. And as it turns out, once the FDA approval was given for implanting those devices on the left, that's just the way it's been done ever since, even with newer devices.
So in reality, it probably wouldn't, as long as the recurrent laryngeal nerve problem wasn't a problem.
Yeah, I think in reality, we would have to do more clinical trials. We now
Right.
know the effects of implanting a device from a company that I founded in 2007 called SetPoint Medical. SetPoint's medical device is about the size of a multivitamin. The whole thing sits on the left vagus nerve. And in 242 patients with rheumatoid arthritis, the primary endpoints showing significant reduction of disease severity were achieved in that trial, which led to its FDA approval to be implanted. So now, because that trial was done on the left, would it work if the device was implanted on the right? That would require another trial.
Right. Right. No, I understand. Is there some difference in the innervation pattern of the recurrent laryngeal nerve from the right side to the left side, or is it just
the anatomic path is different because the recurrent laryngeal nerve gets dragged down by the development of the heart and the aortic arch and subclavian arteries.
Okay.
So there's a longer path on one side versus the other.
Well, I knew that I knew there had to be kind of a reason, because you mentioned one of the, I guess it was one of the side effects is sometimes it can affect the voice box, the vocal folds.
Right. So if you look at the historic literature of what I call Gen 1 vagus nerve stimulators, these devices are sort of a can and lead device, like a cardiac pacemaker. There's an IPG, a pulse generator that sits under the clavicle, and then there's a wire that is tunneled under the skin up to the neck, and then there's a helical coil that wraps around the vagus nerve on the left side in the carotid sheath. That is those devices, those Gen 1 devices, have been used since the 1990s, and we have published follow-up on the side effect profile from at least 250,000 patients over 30 years of follow-up. I estimate that millions of patients have actually been implanted with those devices. In the peer-reviewed studies that are published on 250,000 patients, the side effect rate should be well less than 1% for surgical complications, wound infection, or any injury to the nerve. Those devices fire for 5 minutes a day on, and then 5 minutes, they fire continuously for 24 hours a day, 5 minutes on, 5 minutes off. And they deliver current up to 5 milliamps.
Okay.
So it's a fair amount of current, and it's on for a duty cycle, essentially 12 hours a day, because it's on 5 off 5 all day long. Now, when that amount of current is flowing in that Gen 1 device, if the patient is speaking, their voice may develop a vibrato or a buzzing in their vocal, or they may feel a buzzing in their vocal cords if they're not speaking. Some patients actually feel a buzzing in the corner of their lip, because the platysma muscle can get, or the accessory nerve or other nerves can get activated by that amount of current. The SetPoint device is very different. The SetPoint device, as I said, doesn't have a lead. It doesn't have a wire. Its lead is built into the multivitamin-sized capsule, which sits directly on the vagus nerve. So there's less current leakage. Also, because the current amount is not 5 milliamps like the other one, it is around 400 or 500 microamps. So there's much less current. It's sitting directly on the vagus nerve. And most important for your question, it is only on for 1 minute a day. So the experience of the vocal cord tingling or the buzzing sound in the voice, the vibrato in the voice, that's for 1 minute a day. Right. And that's the main side effect. The other side effects are the same as from any surgical incision. So patients compare that side effect profile to the side effect profile of biologics, for instance, that they take for rheumatoid arthritis or IBD, which have serious side effect rates of between 5 and 8% in some studies. And they have black box warnings because they're immunosuppressive, and they're also invasive because they have to be injected. So when you ask a patient about comparing those side effect profiles, patients are going to prefer and have already been preferring the surgical implant.
So if I'm thinking about this correctly, you have basically through the vagal nerve stimulation, you're enhancing a normal physiological response from that innervation of that vagus nerve versus giving somebody a pharmacological agent that could suppress a whole bunch of other things. So this is a much cleaner approach in my rudimentary understanding. Your understanding is correct. And you summarized it very, very well. If you know that, and we do know that we know that inflammation is good for you in the right amount when it resolves. And inflammation is the evolutionarily evolved response to injury and infection, and it helps heal the wound and fight off the infection. And that's all good. Right. Problem comes when there's too much inflammation, and then you end up with conditions like rheumatoid arthritis. Now, think about it for a minute. Over hundreds of millions of years, evolution figured that out and must have built in place systems of checks and balances to restrain excessive inflammation. And these signals in the vagus nerve fibers, and we estimate it's probably 1,000 fibers, give or take, out of the 200,000, have a normal evolutionarily conserved role, as you said, to dampen inflammation, but not to cause immunosuppression. Evolution would not have retained the vagus nerve signals if they caused immunosuppression and were dangerous. Yeah, they would have been eliminated over the course of evolution. The drugs, as you point out, do cause immunosuppression because they block 100% of the molecules that they're targeting. So, for instance, anti-TNF antibodies have helped millions of patients, and there's some good things about suppressing TNF in some patients. However, if you suppress 100% of the TNF, which is a cytokine that's important in inflammation, but if you suppress 100% of it, which is how monoclonal antibodies work, then you run the risk of immunosuppression and now having secondary infections or other complications. Mm-hmm. With vagus nerve stimulation, in all the studies we've done over 20 years, vagus nerve stimulation does not suppress TNF 100%. It suppresses it 50% or 70%. It takes it down from the toxic levels to the normal levels. And so, as you said, it is a natural mechanism, a natural inflammatory reflex that we are targeting with SetPoint Medical's device to treat rheumatoid arthritis. Yeah, no, I think that's very fascinating. And then, I wanted to circle back to the vagus nerve. Everybody thinks about, parasympathetic versus sympathetic function and, basically as far as rest and digest or, release of cortisol when you're trying to run from the tiger. But the fact that you can have a reduction of TNF alpha from vagal nerve stimulation is it's not in the mainstream of that concept, at least, before reading your book and looking at what you've done. So it just seems empirically to make so much sense because before we continue with this great discussion, I just want to take a quick break to acknowledge this week's sponsor. I'm constantly exploring new avenues to enhance well-being and address any health concerns. Today, I'm thrilled to introduce you to an exciting supplement called Stem Regen, which I take daily and genuinely believe could revolutionize your health journey as well. Stem Regen stands out as a game changer in the realm of wellness. Crafted from 5 potent plant extracts, it boasts a remarkable ability to stimulate the release of your body's own stem cells from your red bone marrow. Why is this significant? Well, stem cells are the superheroes of our body's natural repair system. They possess the extraordinary capacity to morph into various cell types, aiding in tissue regeneration and repair. As we age, the quantity of circulating stem cells in our system reduces significantly, impairing our body's ability to heal and rejuvenate itself effectively. Stem Regen steps in to counter this decline by kickstarting the release of stem cells into circulation, thereby bolstering your body's innate repair mechanisms. Once unleashed into your bloodstream, these mobilized stem cells actively seek out areas of damage or injury, initiating the healing process. By incorporating stem regen into your daily routine, you're essentially empowering your body to tap into its own regenerative prowess more efficiently. I firmly believe that stem regen could be a valuable addition to your wellness regimen, especially if you're seeking to fortify overall health and vitality, accelerate tissue repair, or expedite recovery from injury or illness. This first of its kind stem cell mobilizer is changing the game in regenerative medicine. Don't wait to unleash your body's innate healing capabilities. Visit stemregen.co/Dr. Steven, that's with a PH, and use the code SBPOD to get a 15% discount off your first order of stem regen. You're upregulating something that needs to, well, or in this case, you're downregulating something, but in a normal manner by upregulating the function of the vagus nerve, which is, like you said, it's not eliminating all TNF alpha, and like you have to have some of it. So I look at it as like, it's not carpet bombing, it's specific targeted therapy, which is really, I mean, the optimal for patients.
100% agree. The thing we haven't talked about yet is that the signals are like the brakes in your car. And so when you activate the brakes, you slow the car. When you activate the inflammatory reflex, you slow the inflammation. Mm-hmm. And we've used TNF as an example, but the signals in the vagus nerve that slow inflammation also slow the production or the release of IL-1 and IL-6 and many other cytokines. And the way cytokines work to produce inflammation is they can enhance each other's activity and they can enhance each other's production. And we call that synergy. So the effects of mixing TNF and IL-1 together on a cell or in an animal or in a human are much more powerful, much more significant than they're more than additive. They're synergistic. Right. And so when you remove many cytokines to a healthy level, you remove the synergistic toxicity that they have if they're all there at the same time. And this is a significant insight that I think we don't know for certain yet because we've only studied a few hundred patients so far, but I think time will tell because we've seen this in hundreds, if not thousands of animal experiments. I think time will tell that the benefits of vagus nerve stimulation come by its effect on multiple mediator pathways that evolved to reduce the damaging side effects of inflammation.
Mm-hmm. Very interesting. So are you guys FDA approved for this? I know it's supposed to be coming through any time. Did you get the approval yet?
The approval was announced. SetPoint Medical will be launching, we'll have a product launch in the coming weeks or months based on the recent FDA approval. Yes.
Wow. Congratulations.
Thank you. The congratulations are not just to me, when you look at
your whole team.
Well, the team in the lab has started the work and then continued it. The team in the investing community that took the risk and invested in the company that I started, SetPoint Medical, in 2007 with my colleague from Harvard, Shaw Warren. The employees of the company that took the risk to join a startup company and then work on it for 18 years. Right. The clinical investigators who took the risk on participating in the clinical trials when we didn't know if it would work. And then congratulations to the patients who, look, we all of us, all the people I just said, hundreds of people, we all signed up to do this because we are interested in it and we love doing it. And we want to, we hope it works and helps lots of people. But at the end of the day, we like what we do. None of the patients chose to have their condition. None of the patients woke up and said one day, this is what I want. But they all the clinical trial participants, they all signed up to do this in the hopes, not knowing it would work, in the hopes that, the world would be a better place at the end of the day. And so congratulations all around, right? Congratulations all around.
No, and it just shows, it reflects at how difficult and how expensive it is to get something like this through the FDA. I mean, you said 18 years, millions of dollars, and lots of heartache and stress along the way. And so it's a big accomplishment. And the ultimate benefactors will be the patients, because it's just such, in my mind, it's just such a cleaner way to address this complex problem of the cytokine. I don't want to say cytokine storm, but excessive inflammation and that. So that's very cool. Undoubtedly, this is also going to be used, as you point out in your book, for other conditions. I think you pointed out, I don't know if it's already approved for Crohn's disease or not, but that's one of the things you were looking at, right?
Yes, there have been a series of small, successful clinical trials doing this kind of approach in Crohn's disease and in ulcerative colitis. I expect there will be many more clinical trials in those conditions. I am aware that SetPoint will be pursuing a clinical trial in multiple sclerosis. This is really interesting to me because as you might expect, when you inhibit inflammation in multiple sclerosis, you would expect there to be less damage, as there is in, for instance, rheumatoid arthritis or Crohn's disease.
Right.
But in the animal models that we've looked at so far, the animal models of multiple sclerosis, we did see the reduced inflammation and the reduced damage. What was very surprising is that we also saw increased remyelination. So when in multiple sclerosis, the myelin sheath is damaged, and that impairs the ability of the nerves to signal.
Right.
And when you enhance remyelinization, you raise the possibility for recovery from some lost functions. And if this holds up in clinical trials, this will be a significant advance for multiple sclerosis. But the list goes on. Other autoimmune conditions, psoriatic arthritis, some types of diabetes, potentially even neurodegenerative conditions like Alzheimer's disease and Parkinson's disease. All of these things are either being actively studied or will be studied either in preclinical studies or even in clinical trials. So yeah, there's going to be a lot of new activity coming on this score.
So for these different conditions, you alluded in your book to the gentleman, and I'm not recalling his name right now, who's doing the mapping of all of the different fibers, which is pretty eloquent if you think about it. But for the different diseases, are you using different wavelengths or different amplitudes and frequency?
The study you're referring to is being led by Stavros Zanos, who is my colleague at the Feinstein Institutes in New York. And his lab has a major NIH-supported grant and is part of a consortium creating the first ever complete map of the human vagus nerve, all 200,000 fibers. That's how I know there's 200,000, because he told me there's 100,000 on each side.
Right.
And when you dive into the microanatomy of the vagus nerve in the literature or online, you'll quickly find that there's no map. The maps are all based on sketches from early anatomists in the 19th and 20th centuries. So with modern genetic tools, and molecular tools and neurophysiological tools, Stavros is doing cadaver studies and isolating the vagus nerve 1 millimeter at a time and traveling and following it from its origin in the brain all the way down to each and every individual organ. And
wow.
And then by doing very sophisticated stains, immunohistochemistry, and doing high resolution microscopy, he's able to connect one microscopic slice to the other and build a map all the way from the brain to the liver and the brain to the pancreas and on and on. If you think about how important this nerve is as the major conduit between all the organs in the body and the brain, carrying all these reflex signals that keep your organs functioning in homeostasis, it's pretty surprising that we haven't done this before. Right. So the fact that we're doing it now is perhaps why now? Well, I wish we had done it 20 years ago, but the fact that we're doing it now is actually even better because the tools are so powerful now. You use the modern tools to do this. So yeah, it's a very significant project. Today, Stavros is also working on using nerve stimulation tools such as focused ultrasound or current steering methods to target specific bundles of fibers or small groups of fibers to get a fiber-specific method to stimulate either signals going up into the brain selectively or signals going down to one specific organ or another. And he's making significant progress on this. These are still research tools. They are not in the clinic yet.
But you pointed out in your book with the focused ultrasound how that was and it should be pointed out that it's a little different than the transducer used in a regular sonogram, but you're using that actually much more peripherally down, like over the spleen or over the liver, right? So you're able to stimulate more selectively than if you were going up to the trunk, so to speak.
Focused ultrasound can traverse tissues and can be, as its name suggests, because it's focused, and can be channeled down to a single spot or a single anatomic region, very, very small, a couple millimeters even. If you focus the ultrasound energy on a very small spot like that, you can actually use it to stimulate nerves. And yes, my colleagues Sangeeta Sivan and Stavros Zanos and others at Northwell and at Feinstein have now done the first in human clinical study using focused ultrasound to suppress inflammation. And we did that by targeting the focused ultrasound on the splenic nerve, which is the target of the vagus nerve signals to suppress cytokine release by macrophages and monocytes in the spleen. And it worked beautifully. So if you use focused ultrasound in this way, you can actually, as Sangeeta and Stavros published, you can actually show that there's less cytokine production by white blood cells circulating in the bloodstream of these healthy volunteers. So it raises really interesting questions about developing a new category of specific nerve stimulators that can be placed on the skin or inside the body, but they would work even when they're placed on the skin.
Now, do you see as much of a future for the focused ultrasound because, not because of its less invasive nature, because you're not having to put in an implant, but also because it is, you have the ability to go out peripherally to the splenic nerve or other nerves. Do you think there'll be a big commercial application for that in the future? Or because, I mean, it just seems that that'd be much more cumbersome than putting a small implant in someone's neck next to their left vagus nerve.
I think it's hard to say. What I do know is that to develop any new technology, any new device for therapy takes years and years and years and millions of dollars. I mean, in the case of the set point device, depending on how you count, it costs hundreds of millions of dollars over 18 years. And the reason for that is that you want to build devices that, first of all, you understand how they work, you understand the mechanisms, you do some reductionist science, because you want to make sure you really understand all the nooks and crannies and twists and turns about what you're building, what you're making, how it actually works. And that takes time. And then you have to subject your new device, whether it's an ultrasound or an implant or whatever it is, or a drug, you have to subject it to clinical trials because you want to make sure that it's safe and effective.
Right.
So those things all take time. So anything completely new will take time, money, and regulatory hurdles. The set point device, which is now approved, is the size of a fish oil pill or a multivitamin. It's completely invisible once it's implanted, and it is very safe, and it operates for 1 minute a day. So for people to start a whole new program, I'm not sure what they're competing against at this point, right? We're in a new field with something that seems to work very well and is safe.
Right.
So what are we trying to out-invent? That's kind of the question. I think people will continue to invent new things, and so will I, and that's a good thing. But I think we should stop for 1 minute and say what we have now might be pretty good for a while.
Oh, I didn't mean to imply that it wasn't. I'm just, looking at things, and you always think, wow, you have something technologically, and then there's always evolution after that. So I guess that leads.
Yes.
Yeah.
I didn't take your remark to mean it that way, but I am perhaps responding to many people who I've spoken to who are quick to say, well, what patient with rheumatoid arthritis is going to want surgery? And that is not the right, to me, is not the right question. And you didn't ask it. I'm saying this is what I've heard from other people. And the reason it's not the right question is because the patient is the one who can't button her blouse or can't pick up his pencil because their hands are crippled and painful. They have so much pain they can't go to work, and they're taking medications that are invasive. They have to be injected and cost $100,000 a year and are immunosuppressive and have dangerous side effects. So the real question isn't a trade-off between surgical versus non-surgical therapy. It's a question between risks and benefits of a 1 or 1.5 inch incision in my neck for a 1 hour outpatient procedure that might in fact give me the use of my hands back, and might in fact allow me to stop taking some of these medications. That's the question. And people tend to, when they hear about a totally new approach to therapy, like using a surgical approach instead of a medical approach for a medical condition, they jump to that surgical question and they really don't put themselves in the shoes of the patient, which is, I think, critical, as you know.
Yeah, no. And I can tell you right now, my neuropathic patients, so many of them are so miserable. They'll say, can you operate on me? And it's like, well, no, there's nothing that I can do. Well, try it anyhow. You know what I mean? It's like,
yes,
they're very desperate. But the problem is I think from a non-surgeon perspective, everything that surgery is categorized all the same. Oh, you got to be put to sleep. You have to, all of these different, things that you have to be very worried about. But there's a lot of difference between a small little thing that's very non-invasive and cutting through someone's sternum and cracking it open and doing a 4-level bypass or something like that. So you kind of have to compare apples to apples. And I think a lot of people don't, but I don't think there would be, and my aunt suffered from rheumatoid arthritis horribly. I don't think there's anybody that I would know that wouldn't want to do that, because as you say, it's outpatient. It's a relatively minimally invasive surgery. And I would imagine the recovery time of it is next to nothing.
That's absolutely correct. Let me tell you a really interesting story on this topic. So Set Point Medical, led by the CEO Murthy and the chief medical officer, Dr. Chernoff, that they needed 242 patients in a clinical trial to get the registration trial for the FDA, which was successful, and all the results were announced last November at the American College of Rheumatology, and all the abstracts are available all online, and currently the manuscripts being reviewed for publication. So they needed 242 patients, and they set up a portal, a website for patients to register to be considered for the trial, check for eligibility, all that usual stuff. More than 20,000 patients filled out the whole form. 20,000 patients.
Right.
That tells you the massive pent-up demand by patients. Now, many of those patients, if you ask their physicians, the doctors would say, "Oh, my patient's doing great. They don't they don't want surgery." 20,000 patients wanted to be a participant in the trial.
Right.
And we also saw that in the trial itself, and this will be reported out in the coming months, we also saw that the patients had the option to come into the trial after they had failed to have a clinical benefit from 1 biologic or 2 or even 3. Well, many patients, after the first biologic didn't work for them, said, "No, I don't want a second biologic, even though it's FDA approved. Put me in the trial."
Right. No, and I think that the physicians who say that all their patients are doing well, you have to be a little bit skeptical, because that's not the case, and you have to be like a little bit more realistic when it comes into any type of invasive procedure. And so I think that's just a misguided perception in a way, because what you said there, 20,000 people wanting it. I think the bigger problem is going to be finding enough neurosurgeons to put them in when this thing really hits the ground running. I can't.
You may be right, but I think you and I can both also relate to the other side of the equation. You don't want your doctor, and I don't want my doctor chasing every shiny new object. You want people to have appropriate caution and concern. And so that's really why I wrote the book, The Great Nerve, because I want the information out there for patients to consider the pros and cons and understand the science of it, and for physicians to consider the pros and cons and to understand this. Most physicians haven't thought about the vagus nerve since they were in med school, and that's okay. Right. This is a new thing. When you have something that's really new and different, you actually are asking people to change their workflow, change their behavior, change how they approach to the problem. So, rheumatologists don't normally think about calling a surgeon to help them treat the medical condition of rheumatoid arthritis. It's a whole new concept. And neurosurgeons don't usually think about vagus nerve stimulators in the context of treating a medical condition. So when you have something truly new, people have to think and behave differently, and it requires it requires access to new kinds of information. And I tried to put as much of it as I could in the book, and even the one chapter on FAQs, what the patients could ask the doctor. Right. I mean, the questions might be for the patients, but the answers are for the physicians.
Right. Well, and I don't know. I've always been on the early adopter side of things. So because I've had so many very difficult patients where you're always looking for something more, but as a responsible individual, you're going to take that early adoption mindset still with a level of healthy skepticism, and you're going to want to try to see what you guys are doing and that type of thing. So I don't think it's just jumping on this shiny object in you guys' situation, because you've done this for 18 years. The science is there. I mean, yes, it's foreign. It's a totally different concept. But if somebody takes a pause and actually looks at what you guys have done, they'll say it's empirically good. But I don't see how I mean, even if it doesn't work, it's still not a horrible, experience for the patient. You haven't kept them non-weight bearing for, 6 months or a year for some radical surgery or something. It's like, okay, well, it didn't work. The downside is very minimal in my mind.
I think you bring up two really important points. It's not going to work in everybody. Let's be clear. Right. We have seen some seemingly miraculous recoveries. I've had the pleasure, the privilege, the honor of meeting several patients who have no more signs and symptoms of their rheumatoid arthritis and take no medications. And talking to those people will bring a tear to your eye. I mean, they've sort of gotten their life back. And that's why we all pray that it works in lots of people like that, but it won't be that for everybody. I've met patients and I've read about patients who are a little bit better, but still have to take medication. And then, of course, as in any new therapy, there are patients who don't derive any significant or noticeable benefit, at least in the first 6 months or a year. So we fully expect that the patient responses will bucket into these three groups, and it's going to take implanting many, many more patients before we know the percentages in each group and the risk factors for. If you have been on these drugs or if you have had your condition for this many years, then your odds of being in group A, B, or C will be this, that, or the other. We're a long ways from that yet. We'll have to do studies on many hundreds and thousands of patients to work through all that.
And now a message from our sponsor. I've been taking Nature's Marvels bioregulatory peptides for more than 3 years now on a very consistent basis. It's an integral part into my wellness journey, and I can't imagine not even going on this journey without that. These are designed with cutting-edge science to optimize and restore your body's natural processes. After 3 years, I can say without a doubt, this isn't another wellness product that just gets pushed to the back of the cabinet. This is the real deal for human optimization and longevity. Start your journey today with Nature's Marvels. Our website's below. Well, I think some of it probably is dealing with the type of canvas that you're painting on, right? Because every person's different. Their metabolic status is different. Their comorbidity status is different. They have, let's say, their gut microbiome is much better than another person or whatever. So it's not like you're fighting the same fight with everybody. It's an individual that you're taking care of, not just everybody's the same in that subset. So like all RA patients are not the same, I would imagine. So when you do get this big data, that you'll be able to look at some of those things and say, well, why isn't it working in this particular group as well as it is in this group? And it's like, well, maybe, and I'm just throwing this out hypothetically, maybe it's just because they're not, metabolizing sugar and their blood glucose is too high, or who knows? I mean, there could be myriad reasons of why somebody doesn't get the optimal outcome and another person does.
100% correct. When you look at the rollout of any new therapy, what you see is 100 new questions, and those new questions each require a follow-on research study. And when we we call that sometimes phase 4 research. Phase 1, 2, and 3 gets FDA approval, and phase 4 goes on and on and on, leads to new questions, and sometimes leads to new discoveries that point in the ways of yet again new therapy. So in the case of a condition like rheumatoid arthritis, which is very complicated, let's not overlook the fact that there's no consensus agreement on even what causes it. Mm-hmm. If you don't know the underlying cause and pathophysiology of a condition, it's possible that you're dealing with patients who look like they all have the same condition, but they actually have different conditions.
Right.
And we haven't given them different names yet. But where it's going to be very interesting for me personally is to go into future clinical trials, and we talked earlier about in other conditions, but also to go into future clinical trials of changing the settings on the device. So this device that's implanted is about the size of a multivitamin. It has a battery that's fully rechargeable. It has an electrodes that communicate with the vagus nerve. It has an ASIC, a computer chip, and it has an antenna that communicates with the doctor's tablet or iPad. Now, this communication allows the doctor to wirelessly control how the device works. It can fire for 1 minute or 2 minutes or 5 minutes. It can fire once a day or twice a day or 3 times a day. So we haven't even begun to study the patients that did not have clinical benefit from 1 minute a day. Maybe they'll have clinical benefit from 1 minute twice a day, or 1 minute twice a week, or 1 minute 3 times a day, or 5 minutes 3 times a day. We haven't even begun to explore that yet in clinical trials. Right. And so this is the beginning of a whole new sort of era in this. And the answers have led now to FDA approval. And what you said before was really important, because we've been studying this in the laboratory dating back to the late 1990s. We really do understand a great deal about how it works in mammals, mice, rats, dogs, pigs, and humans. So we have a great deal of knowledge about how this works. In some cases, actually, we understand how these mechanisms and nerve signals work to stop cytokine storm better than we understand how some drugs work, actually. Right. So the opportunity is going to be there for a tremendous amount of new knowledge because of all the work we've done over the past 18 or 20 years on the previous knowledge.
I can imagine that you probably will get some data in these rheumatoid arthritic patients that other comorbidities may be improved or ameliorated. And that'll be kind of interesting to see that, because like you say, the vagus nerve is a homeostatic master circuit. It's kind of like regulating everything. So if you're upregulating the function of the vagus nerve, to me, it would seem that you might get these unexpected great side benefits too.
Yes, I think we're going to see tremendous benefit from reducing the side effects of too much inflammation. I think that's going to be the predominant effect. And the reason I focus on that is because by putting in 400 microamps of current, that's only enough current actually to stimulate the fibers that are exquisitely sensitive to being stimulated. So what does that mean? So in the 100,000 vagus nerve fibers on each side, you have a range of fiber sensitivities to being stimulated. So we put in pulses of electric current, square waves. And these pulses actually activate the nerve to transmit its own electrical signals, which are called action potentials. It's not that we're putting in electric current and it's flowing up and down the copper wire. We're putting in pulses that cause the nerve fibers themselves to send their action potentials traveling up and down within their own fibers. Now, because that's what we're doing, and because we're only putting in a small amount of current, we're not stimulating all 100,000 fibers.
Right.
We're stimulating mostly the fibers that inhibit inflammation. And that means there's fewer side effects and should be fewer off-target activities of this device. But time will tell the complete risk-benefit profile from this after we've done this in many, many more patients. So far, the side effect profile is terrific.
Yeah, that's what you point out in the book. The other thing that you talked about in the book that I wanted to get your insight a little bit on was other ways of vagal nerve stimulation. And you talked about periauricular devices. We used some devices perioperatively, I don't know, 5, 6, 7 years ago, where they would put a little implant in the ear for post-operative pain. And patients did really well with it. Surprisingly, a lot of them took none of their opioids or anything like that. I don't know the mechanism of action, as you point out in your book. That could be vagus, the auricular branch of the vagus nerve, or could be maybe coming off of one of the other nerves like the trigeminal or facial. Who knows, right? But it seemed to have a benefit. What's your take on, because everybody talks about in the biohacking world now, heart rate variability. I'm trying to get more calm. I'm meditating. I'm doing breath work. I'm doing Wim Hof breathing. All of these cool things that everybody is very aware of. Tell me about, I guess, non-implant vagal nerve stimulation.
I think we have to deal with semantics when we talk about this. And let's just start with heart rate variability. Heart rate variability is the variation in time between individual heartbeats that occurs because of something called respiratory sinus arrhythmia. So when you inhale, your diaphragm drops. And when your diaphragm drops, your heart moves a little bit and the pressure in your chest changes. And that means that during inhalation, there is a slight acceleration of heartbeat. And what that does, and you can feel your pulse, and you can take a big inhale and you can feel your heart speed up a little bit. And then if you take a long, slow exhale, your diaphragm comes back up, the pressure in the volume in your chest decreases and the pressure there increases. And that leads to a slowing of heart rate. Now, how does these things happen? When you inhale, signals in your vagus nerve, sensory vagus nerve fibers, transmit the information about the status of oxygen and carbon dioxide and pressure in your lungs during inhalation. So you've stimulated your vagus nerve when you take a big breath in, if you're talking about the sensory fibers carrying information from the lungs to the brain. That incoming information is relayed over to the origin of the vagus nerve in the brain, called the dorsal motor nucleus, or the nucleus ambiguus. Those fibers send signals back down your vagus nerve, which participate in slowing your heart. So you've stimulated during exhalation, if you will, the motor fibers in your vagus nerve, which are that culminate in slowing your heartbeat. And so that's how respiratory sinus arrhythmia works at a very superficial, very, very high level. And that's the origin of heart rate variability. So does taking, 2 or 3 big breaths in quickly and then a long, slow 7-second exhale, is that a vagus nerve stimulator? Well, in this story, you've heard me say vagus nerve activity increases several times, but it's not the same as putting a device on your vagus nerve and driving electrical pulses in. And that's where the semantics kind of break down, because there are fibers in when you exercise, for instance, and you give your muscles a workout and you release norepinephrine from your adrenal glands during exercise and your heart rate goes up. Well, that norepinephrine, that stimulates other fibers in your vagus nerve, which are involved in increasing your heart rate and in your energy level and it's not rest and digest. It's kind of the opposite. That norepinephrine fibers stimulating sensory fibers in the vagus nerve actually increase the output of the locus ceruleus, which is the origin of the sympathetic chain and the fight or flight response. So vagus nerve input can participate in increasing fight or flight output. So just in this little example of heart rate variability and deep breathing and exercise, it gets very confusing to separate one thing from the other. So I have no problem with talking this through. My problem comes when I read something on social media that do this, that, or the other thing to, quote, stimulate your vagus nerve. And I like to say, you have 200,000 vagus nerves. Which ones do you want to stimulate and why? That's where it gets confusing. As we talked about before, there's only 2 ways to specifically or selectively stimulate your vagus nerve. One is to implant an electrode on your vagus nerve, and the other is to use which is now going to be FDA approved for rheumatoid arthritis. The other way to do it is to use focused ultrasound, and that's still an experimental tool. Everything else is either non-specific or perhaps not even truly a vagus nerve stimulator.
Right, right. Did you guys, I think I read this, maybe I'm hallucinating, but you had a small little study among your own folks in the room where I think you're measuring your cytokines before and after some, was it a periauricular stimulation?
Yeah, so I said we should talk about two things, and the first was semantics. So
yeah,
we told the story of heart rate variability and all the different kinds of fibers that are activated during when you take a big breath in and a big breath out, and when you exercise. So semantics are important. The second thing we have to talk about is clinical trial results. Now, if you put, and this has been done by many, many groups and hundreds, literally hundreds of peer-reviewed publications now. But if you put a TENS unit, a transcutaneous electrical nerve stimulator, in the area of the ear, on the cartilage of the ear, outside the ear canal, and that's called the symbiconcha because it looks like a seashell, and you stimulate that region with a TENS unit, there's a good chance you're stimulating the auricular branch of the vagus nerve, which is a sensory nerve, meaning it's carrying that signals into the brainstem, into a place called the nucleus tractus solitarius, which happens to be the destination of all the other incoming sensory fibers in the vagus nerve that goes to your heart and your lungs and your liver and your pancreas, etc. So that's interesting. It's sort of the one place where the vagus nerve comes up to the skin where you can, quote unquote, stimulate it with a TENS unit. But as you pointed out before, there's other nerves to the ear. There's the branches of the occipital nerve, branches of the trigeminal, branches even of the facial nerve. So you don't really know when you put that TENS unit there if you're specifically stimulating the auricular branch of the vagus nerve or some other ones as well, because everyone's, as you know, you're the expert on peripheral nerves. Everyone's peripheral neuroanatomy is a little bit different than everyone else's. There's overlap in the fields. But putting all that aside, let's just say, call it whatever you want. I like to call it transauricular nerve stimulation because I'm not really sure what nerve I'm stimulating, but I am putting electricity into the nerves in the region of the ear. Now you can do a clinical study and you can ask all kinds of interesting questions. If I do this and measure the ability of white blood cells to make cytokines before and after the nerve stimulation, what happens? Well, when Sangeeta and I did this, we showed very clearly that the TENS unit in the ear tended to reduce the amount of inflammatory cytokines being made by the white blood cells before and after. That's fascinating to me. Is that the same as vagus nerve stimulation? Well, it looks quite similar to what we do when we implant a vagus nerve stimulator in the operating room, but it doesn't mean that's the same mechanism. You know, correlation does not prove causality. So that kind of logic can be applied to many, many of those hundreds of clinical studies of transauricular nerve stimulation. And the way I like to break it down is, was the study well designed? Is it statistically powered? Is it the appropriate controls? Is it a big enough population? And can it be replicated? That's a clinical trial strategy question or approach question. It doesn't prove or disprove that transauricular nerve stimulation is a vagus nerve stimulator. Do you follow?
No, I definitely follow you. You know, and I think you pointed out in your book that the one gentleman didn't get a reduction in his cytokines, but had to pee right away. And then you readjusted it so that you got a little bit different location. And then it was, he got a reduction of cytokines and didn't have to go pee right away.
It's 100% correct. It's a fascinating story because it drove us to the acupuncture maps that you can pull up online.
Right.
And the acupuncture map labeled bladder is right next to the part of the ear labeled spleen, which is a fascinating story in and of itself, because take those acupuncture maps for what they're worth and check for yourself of how they could be validated, how the spleen point could ever be validated, escapes my ability to understand. But in fact, the spleen is the place where the vagus nerve signals do inhibit the cytokine production, and that is very close to the place that we were stimulating. So there's a lot of work to do in this space. I mean, we should dispel your listeners of the notion that these ancient acupuncture maps were all mapped out thousands of years ago, because as you know from the book, they were actually discovered and first map by a Frenchman in 1957, but that's
Right.
a longer story in the book.
Right. No, well, I think that it's very interesting. I think what you said about causality versus causation, or correlation versus causation. But when you look at these subsets, it gives you some decent information. It may not be pure as far as an academic arm of a study, but if you're able to stimulate a certain area, and yeah, granted there is a innervation crossover, but the patient still gets better. There's still some merit in looking into that. But I think, what you're saying is that when you do great science, you get more questions. That's
100%.
really what it's
Yes,
about.
it's about answering the questions in a way that we can then apply to the next patient, right? That's what we all want to do. And unless the studies are sufficiently powered and the controls being appropriately randomized in the appropriate control population, sometimes it's very hard to answer that. So, but it's interesting. Now, the third thing I think you have to address when thinking this through, you have to address semantics. You have to address clinical trial design and replication. The third thing you have to address is that if a patient has damaged vagus nerve, which we saw in COVID. We saw it in COVID victims who died of COVID. And this was a very important study, came out of Spain. And we saw in some early studies from what they call long COVID patients who have, by fMRI studies and by high resolution ultrasound studies, have damage in their vagus nerve. They found the presence of the SARS virus in some victims who died of COVID-19. And they saw inflammation in these nerves. So if a patient has damage to the vagus nerve, then all of the self-help advice, do more deep breathing, do more meditation, do more transauricular nerve stimulation. If the nerve is damaged or even some of the fibers are dead, it's not going to work, right?
Right.
You're gonna need something else, potentially an implant, or potentially even the implant won't work. So those are the kinds of questions that I'm always very cautious before recommending. We'll just go do this self-help measure and everything will be fine. I think if it was that easy, a lot of people with rheumatoid arthritis would have gotten themselves better by now. There may be other factors going on that we don't completely understand. Now, if you're a healthy person and you want to increase your heart rate variability through meditation and exercise, I'm a huge fan. I do it every day. I don't exercise every day. I try to exercise 5 days a week. I try to meditate every day. I think it's really a good way to keep your heart rate down. I think keeping your heart rate down and your heart rate variability up is a very good thing for long-term health span and longevity. I think that eating a balanced diet and getting enough sleep and being socially engaged and cognitively active, these are all great things and great advice. Do they all increase your quote unquote vagal tone as reflected in heart rate variability? Yes, they do. Does that mean you have less inflammation? Maybe.
Right.
Are they all connected? Maybe. Those are the kinds of studies that we're working on now to try to break down the specific contribution of the individual fibers to these individual mechanisms.
Right. Well, I think once you get that anatomical mapping done, you're going to have even more insight than you have now, just because it's like knowing the highways to a drive that you're going to do. Well, oh shoot, there's like 4 other highways here that we can go down. So I think that's going to pay a lot of dividends for people long term from you guys' research and everything. But well, I guess one last question I have for you. Let's say one of the listeners out there is very severely afflicted with rheumatoid arthritis. How do they find some hope or places where they can get this device?
The company SetPoint Medical is currently now, based on the recent FDA approval of their device, is going to be rolling out a product launch, and it will be first available in specialized centers. As you can imagine, when you have a new technology, you want to make sure that the first centers that do this have surgeons and medical teams and allied health professionals all fully up to speed on doing the therapy and the surgery, because you want to minimize the risk of any side effects and complications. So the product rollout will be at specialized centers. So I would recommend that potential patients seeking this therapy go to the SetPoint Medical website. We've also launched at Northwell Health in New York a Center for Bioelectronic Medicine, and there is a call-in number and a patient navigator that will be available there for patients who want to seek care in the greater New York area. And that we opened, I believe, a week or two ago.
Wow.
And that you can find online.
Very good. Dr. Kevin Tracey, thank you so much for taking the time, sharing your expertise. The amount of your body of work is really intoxicating and overwhelming. And I don't know how you did it and continue to do it, because it's astounding what you've done, all the patents you have, the publications that you have, and, it's pretty incredible. And thank you for that.
Well, thank you for having me on. It's been a real pleasure talking to you.
Thanks so much.
Well, folks, if you are over 40 and you are not using a supplement to augment nitric oxide production, you should really rethink that. This molecule is absolutely imperative to the health of so many bodily functions, not the least of which is cardioprotection. If you have any question on nitric oxide, I suggest that you register with Approved Medical Solutions and begin receiving their educational emails. I never miss a day of nitric oxide supplementation. That's how important it is. Long overdue, Approved Medical Solutions created the only nitric oxide replenishment formula without oxalates. Approved Medical Solutions does not use beet, spinach, or arginine. Approved Medical Solutions is proud to offer our audience their oxalate-free nitric oxide formula. If you are not a healthcare provider, you can still get started by going to approvedmedicalsolutions.com and use the code SBARRETT at checkout, and you'll receive a 10% medical discount savings. You can register to receive educational emails without any purchase. For licensed practitioners, you too can go to approvedmedicalsolutions.com and get registered today. They have unique bundles for all of the full-fledged spelunkers of the Pod of Inquiry. Use Pod of Inquiry at checkout after registration. Here's a little secret. If you order their testing strips and test every patient for a few clinic days, you will see every one of them be deficient. When they see this result, they will want you to start them up immediately. Now, thanks to Approved Medical Solutions, you can give them the best care without the worry of oxalates. I hope you all enjoyed today's show and got some truly empowering knowledge out of it. You can always follow up on anything we talked about in the show notes found at our website, podofinquiry.com. If you think you might have tickled just a few neurons inside that cerebral cortex, please spread the word to your friends and colleagues and tell them that they need to get spelunking. Also, if you can take a minute to leave a review on your podcast provider of choice, and especially Apple Podcasts, that would greatly help the show and be so appreciated, provided it's positive. And if not, then just keep your pie hole shut and stay down in that cave. Let's keep spelunking.
This podcast is designed for informational purposes only and does not constitute any medical or surgical consulting advice or imply a development of any physician-patient relationship. The opinions of guests who are featured on the show are not necessarily the opinions of Dr. Barrett or the production team. This podcast is owned solely by Barrett Medical and Surgical Media, LLC. While the show is highly oriented for physicians and healthcare providers, anyone interested in the improvement of human performance and understanding will find this a welcome goblet to sip from, or guzzle. However, no representation or warranties are made in any way whatsoever on this podcast for any products, techniques, or other things discussed. Invited guests are not vetted by the Pod of Inquiry for their qualifications and may have a direct or indirect financial interest in what they present and discuss on the show. The Pod of Inquiry disclaims any responsibility from anything taken from the show and used personally or professionally. It is a responsibility of the listener to perform their own due diligence prior to the implementation of any ideas, products, techniques, or anything talked about on the show.